Awesome. Hey, Caitlin.
Hi. Question for you. The last lecture that we had on Tuesday, did you post the recording yet?
No.
Okay.
I will try. It's hard for me to post them during the week. It takes me some time.
Okay.
So I tend to put it off until Friday night or Saturday night,
Okay.
but I will get them posted.
I just missed, like, I wanted to go back over. I just missed like the last little part. And I figured if I went back when it was recorded that it would all like refresh and make sense. So that's okay.
Yeah, give me, I have, normally I'm free like on Friday after 11.
Okay.
But I have to go out to Baltimore on Friday, tomorrow.
Gotcha.
So be patient with me, but I will get them up. Let me aim to get them up by, I'll have them up by midnight tomorrow night.
Okay. Yeah, absolutely.
You know what? And Caitlin, I may be able to, depending on what time I get home tonight, I teach on Thursday evenings and Tuesday nights, but I get home earlier on Thursday evenings. I might be able to get it up tonight. Let me just see what happens.
Okay. Yeah, either or, just so obviously before I do like the chapter 2, all that stuff that's due within the 4th, that I can just re-go over it.
I understand,
Thank you.
of course. First, are there any questions or concerns? So what if you remember from the other day, we talked about the reason that carbon is really the basis for all organic molecules. And when we talk about organic molecules, we're talking about the majority or 99% of the molecules that are necessary for life and that maintain life.
In progress.
Oh, just for a second here. they are, when we just have carbon and hydrogen, the carbon is electroneutral. Hydrogen is not very electronegative. And so carbon is really great because it can double bond with itself, and it can make up to 4 bonds with other things, including itself. However, it's a very non-polar molecule. And things that tend to be non-polar are not very reactive. As a result, if it doesn't react, nothing's going to occur. We won't, no reactions that are going to allow for life are going to occur. Mom, I think she wants to go with you. Are you going to bed? I think she wants to go with you. Go get her. As a result, the only way to start allowing for these carbon skeletons to be involved in life-sustaining reactions, we have to add functional groups to them. And the majority of the functional groups, except for this one right here, result in the molecules that they become bound to, result in them becoming polar molecules, or result in them developing acidic properties or basic properties. So their presence in an organic molecule is what gives the organic molecule the chemical characteristics it has. The R group, and I like to think of R as being something random. So the R is any other part of the skeleton. Any other part of the molecule. So in this case, the R group here would be this black one right here. So any other, whatever the functional group is connected to is an R. So excuse me, let me back up. Functional groups can have hydroxyl groups attached to them, which is an OH, an oxygen and a hydrogen. Most common molecules that have hydroxyl groups attached to them are sugars and alcohols. Because of the oxygen, these tend to be very polar molecules. Methyl groups are the only non-polar functional group that we have. They're very important in gene expression because they silence gene expression at certain regions of the DNA. And if you think about it, all of your cells outside of your red blood cells that don't contain any DNA, all of your cells contain your full genome, all of the genes that you have. All of your DNA that you were born with is contained within each of your cells. But each of the 200 or more types of cells in your body have different functions and they do different things. They do different jobs. That's dictated by the DNA. In order for that to happen, some of the genes, because each cell contains all of your genes, some of the genes have to not be expressed in certain cells and be expressed in other cells. the methyl group is what keeps them from not being expressed, or keeps them, from not being, that's what, I'm sorry, making sure I'm saying the right thing, keeps them from not being expressed, silences certain genes in certain cells that allows given cells to have specific jobs. They're also very nonpolar, meaning they're very hydrophobic. Anything that's charged or polar is going to be hydrophilic. Carbonyl group, very hydrophilic. It is not as hydrophilic as some groups, because if you look, it's found in fats, which don't, and triglycerides, which don't tend to be very hydrophilic. But it still has some polarity to it and some hydrophilic properties to it because of the oxygen that's attached to. Carboxylic group tends to be an acid. This is also called a carboxylic acid. Bear with me just for a second here. So carboxyl is also called a carboxylic acid. And the reason it's also called a carboxylic acid is because it can be, it is an acid. Do you guys remember what is the main definition of an acid? Claire, I'm going to call on you because you're the only face I can see right now. What
is the pH is above 7
or below 7, below 7.
below, I'm sorry,
Below
like
7,
5.
yeah.
Yeah, below
And-
7.
And what also, if we, any given molecule, what determines whether it's an acid or a base?
When it is like 7.4.
Blood is 7.4, and so it's slightly basic.
The pH level.
The pH level.
Well, that's true, but what says whether or not, if you look at any given molecule and you stick it in water, what determines whether it's an acid or a base?
If it dissolves or not.
Well, how does it dissolve?
Through the hydrogen.
Say it again.
How does it dissolve, dog?
What do all acids do? Whether they're strong or weak, they do something. What do they do? Strong acids do it a lot easier and faster than weak acids do.
Diffuse?
Don't they dissociate?
Disassociate.
Yeah, they dissociate.
They disassociate, but what do they disassociate into? What every time they disassociate, what do they give up?
Hydrogen.
Hydrogen.
Exactly, they give up hydrogen ions. Bases always take them. They accept them. So we can call a carboxyl is also called a carboxylic acid because it gives up a hydrogen. It will give up this hydrogen right here very easily. So it ionizes to release hydrogens. When it ionizes, meaning to disassociate, like a lot of people were saying, I think of ionizing as the same as disassociating, because the second it disassociates, each thing that has separated when you disassociate, that has a charge on it. So ionizing, meaning to form ions, is the same. Let me also bear with me just for a second here. Ionizing, ionization equals disassociation in water. Let's say in water. Which means formation of ions. Or formation of things, let's say this, formation of things with, of molecules or atoms with a charge, with charges. Formation of atoms, molecule, or let's put it this way instead. Charged molecules or atoms. So ionization is a dissociation in water, which also results in the formation, resulting in the formation of charged molecules or atoms. If you remember, when it gives the hydrogen ions, whenever we call it an ion because it's now got a charge. Do you remember what the charge is on it?
No.
Is it a +3? Is it a -3? What is it?
Is it giving away?
It's a what?
I said it's giving. I'm just trying to think. Like it's giving away.
It takes on, so if you look here, you read this chart right here, the hydrogen ions have a +1 charge. And the result is that this hydroxyl group, when it gives up this hydrogen right here, because it's an acid, it's going to give it up. The oxygen has a negative charge on it. Because
So that's
remember,
because it's giving it, right?
yep,
'Cause it's
and it
giving it
takes
away.
and it takes the electron from the hydrogen ion and keeps it.
Gotcha.
So carboxylic acids or carboxyls, amino groups accept hydrogen ions. So this is a base. And they also are charged because when they accept that hydrogen ion, those hydrogen ions come with their +1 charge, which ends up giving the overall molecule a +1 charge. Since they can bind, amino groups can bind up hydrogen ions, they're considered basic. Phosphates are charged. They're also very polar. And they can release hydrogen ions. So what are they, an acid or a base if they can release hydrogen ions? Remember, ionization and dissociation are the same thing.
Or an acid.
Acid.
They're an acid. So carboxyl groups can release hydrogen ions into a solution. Or phosphate groups can give up hydrogen ions.
Oh my God.
So sulfohydryl groups are very similar to hydroxyl groups, except the oxygen is replaced by an S, a sulfur. And remember, sulfur is also in that category on the top right periodic table. So it's also a fairly electronegative element. And that's why sulfohydryl groups are also polar. So adding different functional groups to different parts of the molecule will change the chemical properties of the molecules. Each one of these hormones, testosterone and estradiol, which is another name for can be chemically changed to estrogen, have the same overall basic structure. This four-ring molecule is cholesterol. Both of them have the overall basic carbon-hydrogen skeleton of cholesterol. However, the change in the functional groups causes testosterone to become male. People that are males have lots of testosterone. And they believe it or not, and men do create some estrogen, but females do not produce estrogen. Testosterone. Females do not produce testosterone. The production of testosterone is what by default creates males versus females. If your body does not start, if the developing embryo doesn't start to secrete testosterone, it becomes a female by default. So two changes in this carbon skeleton, this carbonyl group here and this methyl group right here versus a hydroxyl group right here, results in this being a female hormone and this being a male hormone. So just one example of where functional groups can massively change the chemical properties of a given molecule. Can you find the functional groups in these different molecules? Acetylsalicylic acid is Tylenol. Aspartame is what is found in a lot of diet drinks. So the question is asking you to circle the functional groups that you can find here. Right here, there's a carboxylic acid. There's a methyl group. There's a carbonyl group. Here, there's a carboxylic acid. There's a hydroxyl group. There's a methyl group right there. And with practice, you get better and better at seeing them, I promise. Here's a carboxyl group. Here's an amino group. This right here, we'll talk about this in a moment, but this is a peptide bond. But this by itself is a carbonyl group. This is a methyl group. This is a carbonyl group. Down here, DNA, deoxyribonucleic acid. It's an amino group. There's a carbonyl group. There's a methyl group right there. There's a phosphate group right there and a phosphate group right there. Phosphate group right there. Phosphate group right there. Get so that you can recognize them. And the best way to do that is first memorize this. And then you'll start to see them. So biomolecules are carbon skeletons that have attached functional groups to them. And depending on what biomolecule they are, they'll have different functional groups attached. So most matter in the body is one of 4 types of biomolecules. The 4 types of biomolecules are proteins, lipids, nucleic acids, and carbohydrates. We also call, a lot of times we call carbohydrates sugars, nucleic acids, DNA and RNA. Lipids, another name that we call this, are fats. So the 4 major organic compounds that comprise our body are what? Oh. The major organic compounds are biomolecules. This word right here, major organic compounds, same thing as biomolecules.
Would it be B?
That's
B.
B.
I think you're right. I think it's B.
Ah,
So the first type of biomolecule are carbohydrates. Carbohydrates means hydrated carbon. So in this group, we're going to have a lot of hydroxyl groups. This group contains sugars and starches. Always, or very close to it, any carbohydrate molecule is going to have a ratio of carbon, hydrogen to oxygen in a 1:2:1 ratio, where there will be 1 carbon to every 2 hydrogens to every 1 oxygen. If you're given on an exam a molecular formula of a molecule and it says, what is this? If it has a nitrogen in it, or actually, let me not say that. Let me put it this way. If it has carbon, hydrogen, and oxygen in a 1:2:1 ratio like glucose, if you look down here, the molecular formula of glucose, 6 carbon to 12 hydrogen to 6 oxygen. If you look for that ratio, if it's approximately 1:2:1, you know it's a carbohydrate. It doesn't matter how long and how complicated it gets, how many, it is a carbohydrate. Here, get rid of the S on maltose. Maltose has 12 carbon, 22 hydrogen, and 11 oxygen. And the reason that this is missing 2 hydrogen and 1 oxygen from that 1:12 to 2:1 ratio is because we've removed the water to connect these two together. Remove the water to connect these two together.
So if I may ask, so what ratio is maltose?
What?
What ratio is maltose?
I'm sorry, say it one more time, Toyin.
I said, what ratio is maltose? Maltose
What sugar is maltose? Maltose,
ratio.
say
Is
it
it
one-
2:1?
it's close. It's very close. If you think here, this is one subunit. Remember we talked about monomers and polymers in the last lecture? This is one monomer right here. This is 2 monomers that have been connected together. And the way we connect them together is we remove a water. And water has a molecular formula of H2O. So if we add, the only difference between this 1:2:1 ratio here and what you'd expect a 1:2 ratio to be, 1:2:1 ratio to be, is 2 hydrogen are missing here and 1 oxygen is missing here. Because we've removed the water here to bind these together. Is that clear? Kind of?
Yes.
Okay. Glycogen is the way we store energy in animals, such as humans. Stored in our livers and our muscle is our way of storing glucose. In plants, it's stored as starch. So the energy storage in humans and other animals is glycogen. In plants, it's starch. So monosaccharides, let me give you some new words here, just a sec. Monosaccharides. Monosaccharides, or so mono means 1, di means 2, tri means 3, and so forth. Saccharide means sugar or carbohydrate. Actually, let's not say sugar, let's say carbohydrate. Or actually, we'll put in sugar too. But the scientific term is carbohydrate, sugar. So anytime you see the word saccharide, it's referring to a carbohydrate. Another one, let me put one more in here just in case. Glyco, like in glycogen, is referring to a carbohydrate. Every time. Remember this glyco for a later part of the class. Glyco always means carbohydrate. So glycogen is a carbohydrate complex. This in mind. monosaccharides are the building blocks. So these are the monomers of carbohydrates that are composed of 3 to 7 carbons. Pentose, oh, let me put another word, bear with me. I'm going to put in another word here. Anything that ends in OSE is a carbohydrate also. So glucose, lactose, maltose, sucrose are all carbohydrates. And you can tell just based on how the last 3 letters. Oh, you can't see my screen yet, just a sec. So pentose is 5 carbons. Hexose is 6 carbons. These are the most important in the body. Some carbohydrates are isomers. Remember isomers? What's an isomer? It's an isomer.
Isn't it a compound that has the same number of atoms?
So it has the same number of atoms. It has the same number of each type of atom, same, but it's got a different arrangement of those atoms. So glucose, galactose, and fructose all have this molecular formula, but the arrangement of the atoms are different. Let me give you an example. Glucose is the universal energy fuel. The brain can only survive on glucose. It can't survive on any of the other monosaccharides. High fructose corn syrup. Oh, let me back up. So glucose is the only one that will easily produce an insulin response. If your blood glucose levels start going up in any normal individual, your insulin levels go up. High fructose corn syrup, which is in many of your juices, unless it's 100% juice, or many of your juices, your Arizona teas and Arizona juices, the next to water, the next ingredient is high fructose corn syrup. It is really bad for you because it doesn't create an insulin response immediately. So your sugar levels start going up, but you can't use it until fructose is converted to glucose. Even though they have the same molecular formula, this does not create an insulin response like glucose will. That's why if you feel like you're getting kind of hypoglycemic, your levels of blood sugar are going down, it does not help you to drink an Arizona drink right immediately. It will not bump up your blood sugar levels. If it does, it's going to take it a lot longer because you have to convert it into glucose. So I kind of answered this already. How might being isomers impact physical and chemical characteristics? If you remember, the first day we talked about how structure was very important to function. Function was very important to structure. If you change the structure of an isomer is going to affect its function. And all isomers may have the same atoms, but they have different functional structures. Any questions? So disaccharides, remember di means two. So these are molecular formulas like that of maltose that contain two hydrogen atoms and one water less than those observed in the two individual monosaccharides, because you have to remove a water to connect them together. So maltose, sucrose, your common table sugar. Lactose, your milk sugars. Polysaccharides, poly meaning more than one. So polysaccharides are usually chains made of similar subunits. Two polymers of glucose. Glucose is a storage molecule. Sorry, starch is a storage molecule formed in plants. Contains a lot of fiber. Cellulose, so we can't use it. It's a good fiber because we can't break it down. When the starch is absorbed, digested, that we can break down, the individual glucose molecules are broken up and absorbed. Glycogen is the storage molecule formed in the storage molecule formed in animals, which is stored in the skeletal muscle and liver, more in the liver than in the muscle. When blood sugar levels drop, glycogen is broken down at its branching ends, and the glucose molecules are released into the blood. They look very similar, and their molecular formulas, they're isomers of one another. But one we easily break down and absorb, glycogen. Starch we don't, not as easily. So if you remember when we were talking about the properties of water, why it's such a great molecule, one of the things that we talked about was that it's involved in hydrolysis reactions and dehydration reactions. Dehydration reactions remove water from two monomers, and they're joined to make polymers. So we remove a water from two monosaccharides and join them into a poly or a disaccharide, make a larger molecule. Hydrolysis, if you think lysis is to break something up and hydro is water, so we add water to break down a larger molecule. With regards to these two reactions, so dehydration, you remove a water to combine two monomers. Hydrolysis, you add a water to break up two monomers. Which of these reactions are anabolic? And which ones are catabolic? Which one is the anabolic reaction here?
Well, cannibalic is like, I don't know why it sticks in my mind, but like cannibalism. So like you're taking something or you're breaking it down. So would that be hydrolysis? And then
That
the
would be-
anabolic would be dehydration.
Yes, and Caitlin, that's the way I remember it too. Is if you think cannibalism is kind of a graphic thing, but hey, it works here. Catabolic meaning to break down, anabolic to combine. Which of these reactions tends to release energy and which one tends to be used to store energy?
Would hydrolysis release energy because you're breaking it down? And once you break something apart, it releases everything?
Anytime, yes, anytime that you have chemical bonds are a form of potential energy. If you break that bond, you release energy. So a dehydration reaction would be a way of storing energy. A hydrolysis reaction is a way of releasing it. So the major function of carbohydrates in the body is what?
B,
Say it one more time, Kevin.
B as in boy.
You're right. B as in boy. It's a cellular fuel. Which type of reaction occurs when biological molecules are broken down?
Hydrolysis, A.
Hydrolysis A. If you remember, one way to remember that hydrolysis is to break down is lysis. If you've ever heard of someone lysing a pimple, lysis is to break down. Lysis, let me put it another way. Lyse. Is to break down. So you're adding in the water to break a bond. Or just not break down so much as just break. Adding the water to break it. So lipid molecules are non-polar compounds. So like carbohydrates, lipids contain a lot of carbon and a lot of hydrogen and oxygen. However, the ratio of oxygen is a lot less. So they still contain carbon, hydrogen, and oxygen, but a lot less oxygen than the other two elements. 4 different types of lipids are triglycerides or neutral fats, phospholipids, steroids, or eosinoids. I think eicosanoids, I think that's how that's said. Triglycerides are called triglycerides because they're composed of 3 fatty acids and 1 glycerol molecule. And like the other synthesis reactions, to combine a fatty acid with the glycerol molecule, you remove a water dehydration reaction. To break it up, you add a water. Remove a water, you combine them. Remove a water, combine them. Remove a water, combine them. So every time in a dehydration reaction, one of the byproducts is going to be water. In all hydrolysis reactions, one of the reactants has to be water. Triglycerides are the most efficient and compact form of stored energy. They store a lot of energy when oxidized, they provide a lot of energy. If you think oxidized, ox meaning to add an oxygen. So best way that I think, we've talked about oxidation, which is to lose electrons. But another way that you can think of oxidation is that you add a lot of oxygen to it. Triglycerides are the most efficient and compact form of stored energy. Deposits of triglycerides can be found under the skin or subcutaneously, where they insulate deeper body tissues. They can vary in their chain length and level of saturation. We have saturated fatty acids, and then we have unsaturated fatty acids. The difference between saturated versus unsaturated is saturated have only single bonds between the carbons. So here we have a carbon, 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 and carbon. And at each of those carbons is completely saturated with as many hydrogens as it can have, minus the two bonds that are, it's got two bonds that connect to adjacent carbons and two bonds that connect to hydrogens. That's the way you know it's saturated. If there is a double bond between two carbons, it's unsaturated because not all of the binding spots that are available for hydrogen are filled with hydrogen. One of the binding spots is being used to make a double bond between the two adjacent carbons. So saturated meaning all of the binding spots that were available to bind to hydrogen, which are two, are bound to hydrogen. When it's unsaturated, we have double bonds within there. We can have monosaturated fatty acids, which is where there's one double bond. Or we can have polysaturated or polyunsaturated. I'm sorry, monounsaturated, polyunsaturated. Polyunsaturated meaning we have more than one double bond. Omega-3 fatty acids like fish oil have multiple double bonds. With regards to these double bonds, we can have covalent single bonds where along the plane here, here's the plane of the double bond. The two connections, actually, let me do this differently. Okay, so with a double bond, with carbon, with a carbon, have a double bond here. And then we have a hydrogen and a hydrogen. This, a lot of times all the time, when you have single bonds, you can rotate things around single bonds. You can rotate if we have bear with me just for a moment here. If we have a single bond here, we can rotate around this bond right here. And we can make, if this could be CH3, this could be CH3. And we can rotate it so that the both of these CH3s are on the same side of this bond. On the same side, or they're on the opposite side, like they are in this case. One methyl group's on this side, one methyl group's on this side. However, once we put in a double bond, like this, it's stuck. We can't rotate it anymore. We can't rotate, we can't flip this methyl group so it's now on this side and this hydrogen here is on this side. This is what we call trans. A trans double bond. And the way that I like to remember trans is that I have to transfer this group to the other side, or the one group is transferred to the other side. So this is a trans. Once I've reinforced the carbon-carbon bond with a double bond, I can no longer rotate this around. So that's a trans where they're on opposite sides of the double bond. Oh, why did I just erase it just so I could draw it again? I don't know. But so this is trans. this is cis. Because now both of these groups are on the same side. Both of these groups, both hydrogens are on the same side. This is cis. So trans is opposite, cis is the same side. So cis, same side. Trans, opposite side. Opposite sides. So trans fatty acids have at least one bond in a trans position, one double bond in a trans position. These trans fatty acids, so in the fatty acid tail, carbon hydrogen tail of a fatty acid, resist enzymatic breakdown in the human body and remain in circulation longer, blood circulation longer, and leave deposits in arteries, thus raising the risk of heart disease. Do you know atherosclerosis is a major killer in our society right now? And lots of people, I don't know, I think they estimated that easily, what was it? 70% of our society deals with atherosclerosis, whether they realize it or not. Cis fatty acids, at least one bond are in the same angle. So they're in the same plane. So cis opposite, one's this way, one's that way. Sorry, trans opposite, one's this way, one's this way. Cis, they're in the same direction. Trans ones resist enzymatic breakdown, making them hard to eliminate and therefore being risk for causing heart disease. Cis, much easier to break down. So are cis and trans fatty acids saturated or unsaturated?
Would it be saturated because it's a
No.
single bond?
Actually, they're double bonds.
So if they're double, it's unsaturated.
Correct. Phospholipids are modified triglycerides that are diglycerides. So remember, a triglyceride is a glycerol molecule attached to 3 fatty acids. A diglyceride has 2 fatty acids attached to the glycerol molecule. Don't you dare, buddy. Don't step on my computer. And 1 phosphate is attached to the last carbon of the glycerol molecule. And a phosphate group. This is one of those functional groups. This is a very polar charged molecule. The fatty acids are very nonpolar. Therefore, triglycerides phospholipids tend to have a polar side of a polar side and a non-polar side to their molecule, making them amphipathic or amphiphilic. When in water, they organize so that the phospholipids are the phosphates groups are the only ones exposed to the water. And the lipids are sequestered inside. So the hydrophilic heads, the phosphates, are exposed to the water, and the hydrophobic tails, the fatty acid tails, are sequestered inside. This is the chief component of our cell membranes. Any questions so far? Steroids are fat soluble flat molecules made of 4 interlocking rings. Right here and right here. Anytime that you see the 4 interlocking rings like this is a cholesterol molecule. It's precursor. Anytime that you see this on any molecule, that molecule was derived from cholesterol. It was made from cholesterol. So cholesterol is a precursor for all steroids, like testosterone, like estrogen, like estradiol, like cortisol. They're all made from cholesterol. So we have good and bad cholesterol. We need to have a certain amount of cholesterol in our body because we have to have what we need to make steroids. We also have to have a certain amount in our cell membranes that allow for a good fluidity of our cell membranes, which we'll talk about more in the next chapter. The public are always referring to droplets in the blood called lipoproteins. These are complexes of cholesterol, fat, phospholipids, and proteins. Low density lipoproteins have a high ratio of lipid to protein and contribute to cardiovascular disease. So they have more lipid than they do protein. So they're low density lipoproteins. High density lipoproteins have a low ratio, so they have more protein than they do lipid and can help prevent cardiovascular disease. Atherosclerotic plaques are low density lipoprotein accumulations. And low density lipoproteins store cholesterol in the bloodstream. High density lipoproteins regulate low density lipoprotein storage and promote their breakdown and excretion. So a lot of times if you think high density lipoproteins, they're the healthy ones. Low density because of their high levels of lipids, knowing that cholesterol is one of our lipids, high density of lipids to protein, which is why it contributes to cardiovascular disease. I don't know how to say this word. I think it's eicosanoids. Diverse lipids that are made from a 20 carbon fatty acid called arachidonic acid. which is found in all cell membranes. It is most important. The most important eicosanoids are prostaglandins and their derivatives. I am wondering, and I'm going to look it up really quickly here. Bear with me just for a second here. I'm looking it up. I want to know, because if you look here, it says it functions in blood clotting, inflammation, and labor contractions. I want to know, if you remember, we talked in the first chapter, we talked a lot about oxytocin. I'm wondering if it's an eicosanoid. It is involved. So eicosanoids are involved in the positive feedback loops that result in labor contractions, because they are involved in releasing, in allowing for the release of oxytocin. Do you remember that one? Oxytocin?
Yeah.
So the three major classes of lipids include phospholipids, steroids, and what?
Triglycerides.
Triglycerides,
T.
right?
Yeah.
T. Any questions? So so we've talked about lipids, we've talked about carbohydrates. Proteins are next. Proteins make up 10 to 30% of a cell's mass. They are the basic structural materials and can also be enzymes. All enzymes are proteins. Not all proteins are enzymes. This word right here, enzymes. But proteins make up 10 to 30% of a cell's mass. They are the basic structural components and can be used as enzymes. All of these contain carbon, oxygen, hydrogen, and nitrogen. So if you remember that carbohydrates have the ratio of 1 carbon to 2 hydrogen to 1 oxygen. Lipids have carbon, hydrogen, and oxygen, but they have less oxygen than they do carbon or hydrogen. Proteins have all proteins contain nitrogen also. They may also contain sulfur and phosphorus. The full set of proteins made by the human body is the proteome. If you think genome is your full set of genes, proteome is your full set of proteins. So the monomers for making proteins are amino acids. When they're connected together through the same dehydration reaction as we saw before, we make a peptide bond. And to break up a peptide bond, we use the same old hydrolysis reaction we use for all the others. And so each amino acid within a sequence of for a protein is each one is connected to the next one with a peptide bond that is formed with the dehydration reaction. And there are 20 amino acids that work kind of like an alphabet. So a 20-letter alphabet with linkages between the letters. And different amino acids have different properties. Most proteins are macromolecules with polypeptides. Think poly meaning more than one, peptide bond. Polypeptide meaning more than one peptide bond. So most proteins are macromolecules with polypeptides containing 100 to 10,000 amino acids. Big polypeptides. So each different amino acid is like a letter, and their various combinations are going to give rise to a seemingly endless diversity of words. All right, I think we should stop right there. I know we could keep going, but I think that's a good stopping point for the moment. Are there any questions?
No.
Okay. I will see you guys next Tuesday.
Thank you.
Thank you.
Welcome.
Thank you.
Bye, thanks.
Bye.
Welcome. Alex, are you there?